Okay, so we had ended by finishing the unit. We had finished Unit 4, finished the PowerPoint. You have your last lab exercise, that's due Thursday. Friday, you have your exam. It opens up at 12:20. There are two parts to the test. There is a Canvas part that has like multiple choice and some like very short fill in the blank type of questions. That's worth 80 points. And the timer for that is set for 80 minutes. It opens up at 12:20. You'll do that part, and then you can go to the essay part. It's 2 essay questions. I'm asking that you download the Word document, you type in your responses, and then you submit it to that Canvas link. What I will do is I'll create a module on the homepage that says Exam 4, and I'll move it to the top of your homepage. So it'll be like kind of big and bold and ready for you to go. Yeah, you have until 2 p.m. to turn in the essay parts, and I'm asking that you submit it through that Canvas link because we'll go through a program Verisite, which will check for plagiarism specifically. So just ask that you do it that way as opposed to emailing it to me. I also am the kind of person that I like to keep my inbox at 0 unread messages, and I start to get anxious as emails pop in. And so keeping it all together will help me from losing your email or getting a bunch of emails all at once, and it will just, yeah, it'll be better. We'll put your essays in that Canvas box. So multiple choice fill in the blank section worth 80 points, and then essay section worth 20 points on Friday, starting at 12:20. You can finish by 2 p.m. Is the essay based on only on the material for this test, or is it more? No. So there's no, Dr. Pogan and I had discussed it, and we got rid of the cumulative exam. So it's all Unit 4 stuff. It's coming from the materials that I have provided you. So that is the PowerPoint, the lab handout, the lab activities, some of the activities I've made you do, some of the discussion questions we've talked about, and then the textbook readings that I've assigned. That is where the material is coming for this exam. Yes, that's a good question. Yeah, it's not going to be, not making you go all the way back to Unit 1. Will there still be extra credit questions? Not at the moment in its current form, I don't think. There might be one. I would have to go back and look. I don't think there are extra credit questions in its current format. Yes, I do curve. So once everything is done, I will look at it. I will curve it. I have to warn you that as with you guys are taking a bunch of exams, I'm also grading a bunch of exams, and so I'm going to do my best to get things graded in a timely manner. I think I've done that so far this semester, and I'm going to try to do that. I'm a little swamped with exams. I know you have discussion questions that I owe you answers for, or grades for. Again, I'll be just grading that based on whether you did it or not. So what I probably do is throw up the grades for that, and then if you have a question about an individual one of those discussion questions, if something from reading your answers and reading your peers' answers still doesn't make sense, the best thing you could do for that is to actually shoot me an email, and I'll answer your question over email, or we can set up a time to WebEx individually, and that would just save me from going through and responding to everyone's question or responding to everyone's discussion posts when you probably have, through doing it and from reading other people's, kind of figured out the answer along the way. So we'll do it that way. Are there any other questions about the format of the test? Okay. I'm going to go, while you think about it, we can percolate on that. I'm going to switch to the PowerPoint. Okay. Keep the chat up. Slideshow here. Okay, so Unit 4, renal physiology here. I got not a ton of questions, so I'm just going to go through the ones I have. If you feel like I didn't answer your question, you can chat it, or you can also shoot me an email at the end, and I'll answer it that way as well. These I felt like were the most pertinent of the questions that I had gotten. So the first is question like, what does the distal tubule do? So we went through and we talked about the parts of the nephron, right? We went through what each of them does in particular, and we spent a lot of time talking about the loop of Henle and how it builds and maintains this osmotic gradient in the medulla of your nephron, and how that gradient is super important for pulling water out and helping us concentrate this filtrate. And then I think I might have lost the train, the thought train, as we got all the way to the distal tubule. The distal tubule is super important. It's also sometimes called the distal convoluted tubule. And it is where our bodies regulate the amount of water and salts that we reabsorb and we recover. So along the way, as we move through the proximal convoluted tubule into the loop of Henle, we've been reabsorbing water and salt based on that gradient and just based on the physics behind solute drag and gradients and transporters, right? But there hasn't been any control over it. So it's been a process that happens without the body's input. Whereas when we get to the distal tubule, this is where our body, through hormones and through the action of hormones, can regulate the amount of water and salt being reabsorbed and recovered. And you do that in times when it needs to, say, take in more water and increase blood volume when blood pressure falls, for example. That is something we've talked about a lot. So those hormones, they act in the distal tubule and later in the collecting duct. So things like our mineralocorticoids, antidiuretic hormone, PTH, parathyroid hormone, they act here in the distal tubule. And so this is where we're going to reabsorb most of the remaining sodium chloride through some of those epithelial sodium channels, for example. And we can also change the expression. Remember, we talked about with antidiuretic hormone, we can change the expression of aquaporins. When we don't have enough water, we can recover more water. The distal tubule is also where we're going to secrete potassium. So bringing potassium back from the blood plasma into the filtrate. So it's got this potassium secretion and then control with regards to sodium and water. Collecting duct also has that regulatory capacity. We have principal cells and intercalated cells. Principal cells are going to secrete potassium as well, and they're going to reabsorb sodium. And then the intercalated cells respond to acid-base balance and use that acid-base balance to couple it to potassium import. Now, we didn't talk about acid-base balance when the schedule got, whoosh, all messed up, and we lost a week of school. I looked at the topics that I was going to cover, and I was like, acid-base, pH, get out of here. I'm not talking about it. So I'm, that part of the distal, that part of the collecting duct, rather, we're not going to focus on too much. Okay. The other major questions that people had were about the RAS system. So how do we control our filtration rate when our blood pressure changes? Remember, filtration rate, we're talking about things moving from the glomerulus into Bowman's capsule, things moving from the plasma into the filtrate. And at that step, everything is going to move except for large proteins and cells. Big things will stay, everything else will move, and then our body will pull back what it wants. So the metaphor I heard was like cleaning out your fridge. To clean out your fridge, you take everything out of the fridge, and then you just put back what you want, and you throw out the dirty stuff. Our body does the same thing. We're going to take everything out of the blood, we're going to put back what we want, and we're going to throw out the waste here. So we want to maintain the rate at which plasma is filtered, the mils per minute that we're filtering that plasma, that a substance is leaving the plasma. We want to control that so that it stays constant, no matter what happens upstream, downstream of the glomerulus, whether the afferent arteriole is sending a bunch more, right, high blood volume, high blood pressure. We want the GFR, the filtration rate, to stay constant. Okay, so that's our body's goal. Now, there are two ways to do that, two categories, if you would, of ways to do that. Intrinsically, kidney itself does it. Extrinsically, we got hormones flying around. Intrinsically, kidney itself. Extrinsically, hormones flying around. Intrinsic is like when you have a very small problem and you don't need a lot of help and you can just kind of like fix it yourself. So changing the windshield wiper in our, changing the windshield wiper fluid in our car, small problem, I can do that. I'm capable of that small change. If we have a big problem, that's where we need to have extrinsic. Okay. Dr. Pogue is here. If anyone has any questions for him. Any questions for Dr. Pogue while I take a pause and drink a little water? You can put them in the chat. Once. Twice. Okay. I'm going to keep talking about kidneys. So, intrinsic, small change. Extrinsic, we got a big change, we got a big problem. So we talked with intrinsic regulation about a few different ways that our body responds to small changes, right? So we have the myogenic regulation. We're going to have an increase in that arterial pressure, going to activate stretch receptors. That's going to cause this depolarization in that smooth muscle, and that's going to vasoconstrict. And that will bring the filtration rate back down to baseline. Okay. We also can have our tubulo-glomerular feedback, which will change the resistance at the arterial. So the macula densa cells, those important cells in the distal tubule that we talked about, will sense an increase in pressure, and they'll send out a chemical signal that will constrict the arterial again and decrease that pressure. So small things that can happen. Macula densa, again, this is that complicated juxtaglomerular apparatus area where we got arterioles, we got the glomerulus, we got this distal tubule with these specialized cells all coming together. Make sure you have a picture of that in your head when you're moving forward. Okay, the mesangial cells themselves, they help regulate that filter, and so they can change the filtration rate by changing the amount that filters, whether the filter is open more or open less. And this is independent of the vasculature. Okay, that works again over a small window, a narrow range. So you got a small problem, you can fix it yourself. You got a big problem, you're gonna have to call in a mechanic. You're gonna have to call in help. So when we call in help here, we're gonna use hormones. And this is when we have a change in blood pressure that is a lot, or it's over a long period of time. So you're super dehydrated, you have a lot of blood loss, big things are happening, and you're going to use endocrine hormones to help you increase your blood volume and bring your BP back. So for this, we have that renin, angiotensin, aldosterone system. And so first I described it in words, then we went over it in pictures, and then we broke it down. So in words, first, we have those JG cells are going to release renin with the end goal again, is to increase our blood volume and increase our blood pressure. So the JG cells have different triggers. They can either sense the low blood pressure, they can be activated by our sympathetic nervous system, right? Fight or flight, or the macula densa cells can sense sodium issues and then signal to the JG cells nearby. They will release that renin, and the renin will go on to activate angiotensinogen from the liver to make angiotensin 1, which is cleaved by ACE, an enzyme that will make us angiotensin 2. And angiotensin 2 has wide-reaching effects, right? It's going to vasoconstrict, it's going to affect the kidneys themselves, and it's also going to signal to release ADH and aldosterone, which will have even more effects, again, with that end goal of increasing our blood pressure. This is the picture that I came back to, as opposed to the scary picture in your textbook with all the boxes. So again, if we start at the beginning here, we have the liver releasing angiotensinogen, we have the juxtaglomerular apparatus, the cells there releasing renin, and that's going to have us convert angiotensinogen to angiotensin 1. And this is happening in our bloodstream, right? So these are things secreted into circulation, moving around, okay? We're going to have ACE. It's going to be released from the epithelial cells themselves, mainly from the lungs, but also from other parts of the body. And that's going to help cleave and form angiotensin 2, which has those wide effects, right? Sympathetic activity. It's going to help with reabsorption of sodium, potassium excretion, water, retention. And then it's going to have a vasoconstrictive effect itself. And then it will signal to the adrenal gland to release aldosterone, and it will signal to the pituitary, or via the pituitary, to release ADH, antidiuretic hormone. All of those together, we're going to increase our blood volume, increase our BP. We go back to renin now that I've done the overview. Let's go back to that first step again. We release from those JG cells, again, when they sense low blood pressure, when they're innervated by our sympathetic nervous system, or when there is low sodium in the filtrate, the macula densa cells will send a chemical signal, we call it a paracrine signal because it's acting nearby, that will signal to the juxtaglomerular cells to release renin. And that's shown here. Again, we got sympathetic activity, decrease in pressure, decreased GFR. We have a decrease in sodium then in our distal tubule, which will have a paracrine secretion, will cause the release of renin. Renin, again, will start that chain of events, and we end that chain of events with angiotensin II, which has diverse effects. Okay, it does things itself to tubules. It vasoconstricts. Oh. It does things itself. It's vasoconstricting. But importantly, it triggers the release of aldosterone and ADH, which also have diverse effects, which we talked about. So again, ADH. This is coming from the pituitary, from a signal from the hypothalamus. Hypothalamus is going to take in information, either angiotensin II, sympathetic nervous system, hyperosmolarity itself. All of that will signal to the release ADH, which is also known as vasopressin. It's going to go to the kidneys, help us increase our blood volume. Okay. ADH acts on the aquaporins. So what ADH is going to do is when it binds to its G protein coupled receptor, then we're going to have that cyclic AMP to protein kinase A pathway, right, that we've talked about before. That will have vesicles that are already primed and ready to go with aquaporins bind and fuse to the membrane, and will increase the expression then on the membrane, the surface expression of our aquaporins. So we can move more water, we can cover more water. Hypothalamus is involved here. Remember, hypothalamus leading to the pituitary. And this is where we can have our body to feel thirsty. So one way to increase your blood volume, drink more water. That's going to be through the hypothalamus. Okay? They have their own osmolar receptors. They're going to detect the osmolarity, and they're going to signal it's time to drink water. The other downstream from angiotensin II, we have aldosterone. This is the mineralocorticoid secreted by the adrenal cortex. It's going to stimulate sodium reabsorption. Remember, water will then follow that sodium. It's going to excrete more potassium. It's trigger again angiotensin II, or it can also be triggered from the adrenal cortex with high plasma levels of potassium. And then it's going to target the distal tubule, the collecting duct. And this is a steroid. So you need to think about the biochemistry of steroids and where they act, right? Because they can pass through those plasma membrane, and it's going to, instead of affecting phosphorylation cascade, it's going to affect transcription. And so the time frame for this is going to be much longer. Okay, so this shows that, again, affecting transcription. And those targets are things that are going to help with the movement of salt. So we're going to have an increase in the sodium potassium ATPase on the basolateral membrane of our principal cells. We're going to have more potassium and sodium channels, and what it will be is a net exchange of plasma, potassium for sodium in the urine. No effect directly on water. So unlike ADH, which affects the aquaporins, directly affects water, aldosterone is an indirect effect. On the reverse, counteracting this, we talked about AMP, atrial natriuretic peptide. So produced by the atrial cells in response to high blood volume. So the other system with the system we just talked about, the RAS system, is if we want to bring BP up. Well, what happens if we want to bring BP down? That's where AMP is going to come into play. It's going to target the kidney. We're going to switch it around. We're going to excrete, get rid of the sodium, and we're also going to increase blood flow, relax those filter cells, and inhibit aldosterone and inhibit renin. So turn down that RAS system that we just talked about. And all of this is a review. If you're a picture person, here's the same thing in pictures. Okay, so that was our lightning fast review based on questions I had received. Are there other questions people have for me? Questions about the exam, questions about the material. Will the test be like the last one where we cannot go back once we go to the next question? Yes. It has to be like the last one where you see one question at a time and you can't go back. That's a good question. Questions? test. Going once, going twice. Okay, as I said, as a reminder, we have the last bit of lab activity. What is the essay going to be about? You need to figure that out. You will see it when you see it. Yeah, it's I don't divulge that information. If you are structuring your test studying, you want to focus on the material that I focused on. So ideas for what would be the focus, go back, look at the learning objectives, go back, look at the questions I've asked in that discussion question post, the things I've had you do, and that will clue you into the things that I'm focusing on. But again, when you study, it's all the material, right? The PowerPoint, the lab activities, the textbook readings that I've been telling you to read, the homeworks, the activities, a cumulative of this unit all together. Yes. Any other questions about the test? Okay, I'm going to stop the recording, and then you guys are